Now, I assume that most of you are here know about Daniel's work, so he probably doesn't need extensive introduction, but just to illustrate the importance of his work, last night over dinner we were musing about how come the endogenous cannabinoids, which is such a promising new endogenous signaling and ligand system, how come more people are not working in it? And my observation was that probably Daniel, intimidated with his tremendous contributions to the field, many others who are considering moving into this area. He certainly published spectacular work in the last couple of years, and a quick scan of his impressive CV indicates that he has no fewer than 9 papers in Nature, and most of them in the last, I would say, 6 or 7 years, which is certainly an enviable indication of productivity. He also wasted no time after he moved from the protected institute environment into the larger extramural community, and on the first try he got 3 very good R01 grants, which support his excellent work. So today Daniel will talk about, I guess it will be more like a review type lecture summarizing hopefully his most interesting recent findings. And his talk is entitled Endogenous Cannabinoids, Biochemistry and Pharmacology of a Novel Signaling System. Daniel?
Thank you very much, George, for such a flattering introduction. I hope I won't intimidate any of you. I don't think I do, but if I do, please let me know. I'd like to accomplish 2 things today. The first is to do an overview with you of what we think we know about the biochemistry of the endogenous cannabinoid system, and that will occupy the first quarter or third of my presentation. And then I'd like to give you examples taken from our own research of the possible physiological significance of this signaling system. And as I'm doing that, I will also like to highlight the potential therapeutic interest of this system in terms of potential in generating new medicinal agents. Let me start with, may I have the first slide, please? Let me start off by reminding you of some basic pharmacology of the cannabinoid system, the exogenous cannabinoid system. I typically expect cannabis to be well known to most people, so I don't think it does need a whole lot of introduction, but I put up here the plant as it is shown in a 5th century manuscript of Dioscorides Materia Medica. It was known already since then. We know, of course, a whole lot more since the 5th century AD. We know, for example, that this plant contains approximately 400 cannabinol compounds. And of these several hundred compounds, there is one that is particularly interesting and is shown here highlighted, is delta-9 tetrahydrocannabinol, which is considered the main active, or if not the only active, pharmacologically active compound in cannabis, marijuana, and hashish preparations. There are many things that cannabis as well as THC do, and I have summarized them here. This is far from complete, but gives you a fairly good idea of the widespread effects of this class of molecules. Of course, there are the psychoactivity, the psychoactive effects, which are exemplified here in regulation of movement, inhibition of pain, impairments in short-term memory. But there are also a number of peripheral effects that are shown here. For example, regulation of blood pressure, and Dr. Kunos has been one of the pioneering scientists in this area, regulation of intestinal propulsion, Is it better? Okay, sorry about that. I couldn't tell.
peripheral analgesia, participation may be in stress, here in the adrenal medulla, and regulation of bronchial responsiveness. And towards the end of my presentation, as part of my examples on possible physiological roles of the cannabinoid system, of the endogenous cannabinoid system, I'd like to talk briefly about this particular role in regulating bronchial responsiveness. All these effects are produced by activation, by THC activation of a little small group of of receptors, G protein coupled receptors, known as cannabinoid receptors. And these receptors come into main flavors, the CB1 and the CB2. They are, as I said, both G protein coupled receptors, and in heterologous expression systems, they typically couple to inhibition of adenylate cyclase through interaction with GI-GO type of proteins. They share only approximately 44% sequence homology, and they are different also in other ways. For example, the CB1 is most extensively found in the central nervous system, as well as in the peripheral nervous system. It's also present, however, in immune cells and in cells of the reproductive system, and here and there in other cells of the body. So it's fairly ubiquitous by and large. On the other hand, the CB2 receptor appears to be more selectively expressed in cells of immune origin. Attempts to detect it in other cells, particularly in the brain, have been so far negative. And although probably immune cells are not the only source of this receptor, they're probably the most relevant, most important one. And I'd like to, to honor NIH, I'd like to show you one piece of work that was done right here by Miles Erkenham, distribution of the cannabinoid receptors as detected by a ligand, a radioactively labeled ligand here, in a section of rat brain. And you can appreciate both the extensive distribution of this receptor as well as the discreteness. You see, for example, areas such as the coated putamen, the globus pallidus, the intopedunculus nucleus, the substantia nigra pars reticulata that light up extensively. These are all areas involved in motor control, and we know that one of the prominent effects of cannabis intoxication is indeed to affect the motor system. Also the cerebellum, of course, is involved in the control of motor coordination. Likewise, the hippocampus and somewhat the cortex are also heavily labeled. This may or may not, but is currently thought to be one of the neural substrates of the cognitive effects of cannabinoids. So the existence of a cannabinoid receptor and the discrete distribution of this receptor has been baffling for many years, and has suggested the existence of endogenous cannabinoids, as in the case of the opiate receptor and the endogenous opiates. And we have now a small but interesting library of endogenous cannabinoids. They come into, like the cannabinoid receptors, into two major classes. One is called the acyl ethanolamides, and the other one is this guy here, 2-arachidonyl glycerol. First of all, let's start from the top here. Anandamide. Anandamide was discovered by Bill Devane in Rafi Meshulam's lab, 1992. It was published in 1992. As you can see by looking at the molecule of this small compound, it is not the traditional peptide or the biogenic amine or the amino acid as we are used to when we think about signaling molecules in the brain. It is indeed instead a lipid, a fat-derived molecule that contains the structure of arachidonic acid right here and ethanolamine right here. So this is ethanolamide of arachidonic acid. And Bill Devane called it anandamide because ananda in Sanskrit, which is one of Bill's passions, Sanskrit means bliss, and amide, of course, for the chemical nature of the bond here. I don't know if it was his own bliss at discovering it, which must have been in a way because, after toiling for 3 years on this, I'm sure he was happy to find it. But also the fact that he thought that this compound is involved in regulating, of course, mood, which may or may not. We don't know that yet. Anyway, anandamide looks in a way like prostanoids, like eicosanoids, because also it's a derivative of arachidonic acid, and it may share some properties of the eicosanoids, although I don't think it's appropriate to call it as an eicosanoid, but that is really not very important. What is important is to point out that anandamide binds to CB1 receptors with fairly high affinity, not so well to the CB2 receptor, but pretty nicely effective on the CB1. And I put down here two additional molecules, palmitylethanolamide and oleoethanolamide. Let me remind you, if we called anandamide by its chemical name, it would be arachidonylethanolamide, right? So these two compounds here, as you can appreciate by looking at this portion, look very much like anandamide. And the reason why I put them up here is because they are abundantly present, not only in the brain, but in a number of other organs, and they share a lot of biogenetic properties of anandamide. We don't know, however, what the function or functions of these compounds are, if any. We know that they don't bind to cannabinoid receptors. There are some interesting properties that these compounds have. For example, this compound here, palmitylethanolamide, is a potent analgesic, and it may have something to do with CB2 receptor, but the pharmacology is still pretty primitive, and we cannot draw a firm conclusion on this point. The second group of endogenous cannabinoids is exemplified by 2-arachidonyl glycerol, which obviously looks very much like anandamide. Instead of having the ethanolamine moiety here, has a glycerol moiety. It is not very potent on either CB1 or CB2, but is effective and in fact is a full agonist as opposed to anandamide, which is instead a partial agonist. What I would like to do in the next couple of slides is to try and go over the properties, the biochemical properties of these compounds that we think make them unique and interesting. And I think a basic message which I'd like to give at the outset is that there is something basically different about these molecules when you compare them to classical signaling substances such as neurotransmitters or neuropeptide modulators. In classical neurotransmission, we have, of course, a presynaptic bouton delivering when calcium, when an action potential invades the bouton, delivering messengers on the postsynaptic spine, let's say, activating receptors and producing responses. So we have an exocytosis process going on here. With the endocannabinoids, we have a situation that resembles, as I alluded to before, that of the eicosanoids. We don't believe that there is a process of accumulation of the endocannabinoids in vesicles. We do believe that when a primary transmitter is released, activates receptors, which in turn cause the cleavage of membrane precursors in a postsynaptic cell. So this is exemplified here by these yellow sticks here, which are cleaved from this more complex blue and yellow molecule. So once the endocannabinoids are released, they're free to go back into the presynaptic ending or stay postsynaptically to activate cannabinoid receptors, and this will depend, of course, on the presence of the receptor in the respective presynaptic and postsynaptic components. So this, again, this is a mode of action that is dramatically different from classical transmitters, but it's more similar to the mode of action of the eicosanoids, and it's sort of a volume transmission, if you like, or an autocrine effect. Does this mean that these compounds are not interesting? I actually don't believe so, because a lot of exciting pharmacology can be done with modulatory compounds, as I hopefully will be able to show you later on. Okay, on to a more detailed biochemistry, the pathway of anandamide formation. This is by no means a completely accepted model, but I think is fairly accepted right now. And it runs as follows. Let's focus first of all on this guy here. This is the phospholipid precursor. This is the membrane. It's a little dark, but this is the membrane. And this molecule here is the phospholipid precursor, which others and ourselves believe it's the precursor for anandamide. It looks complicated, but it's actually, if I walk through it, we will see that it's very simple. This white portion here is basically phosphatidylethanolamine. But it's linked with an amide bond to arachidonic acid, which I have depicted as bending back into the membrane for obvious reasons of hydrophobicity. There is an enzyme which is still unknown, which we call phospholipase D, because of the reaction it carries out, which cleaves this bond here, the distal phosphodiester bond, forming in a single step anandamide. As phospholipase D carries out this reaction, the precursor, N-arachidonyl phosphatidyletanolamine, or NAPE, we call it for short, NAPE is hydrolyzed very rapidly needs to be resynthesized, and there is an enzyme which does so by moving a fatty acid moiety, such as this arachidonic acid moiety in phosphatidylcholine, onto the primary amino group of PE, forming N-arachidonyl PE. This enzyme is a calcium and cyclic AMP-dependent enzyme called N-acyl transferase, implying this transfer reaction. The formation of 2-AG has the basic molecular logic, but is different in the characters of the play. The precursor is right here is probably phosphatidyl bisphosphate, which is well known to all of us because of its role as a precursor of this molecule, diacylglycerol, or DAG. The reaction is carried out by phospholipase C, which is activated by receptors, etc. Now, that DAG, we know very well, is an activator of protein kinase C, so it's considered a second messenger. And it is a second messenger, but it is also, in our opinion, the precursor for this transcellular messenger to arachidonyl glycerol. And the reaction is carried out by an enzyme which remains, unfortunately, unknown in terms of its molecular structure, called diacylglycerol lipase, which cleaves this moiety here, forming monoacylglycerol, 2-AG. So once a compound is produced and acts, it needs to be inactivated, and it's worthwhile to look at the mechanisms of signal inactivation for many reasons, both basic and therapeutic. As we will see later on, these mechanisms can be target for pharmacological agents of considerable interest. So let's look at the anandamide inactivation, then at the 2-AG inactivation. Anandamide first. Anandamide, against all expectations, anandamide, a small lipid, indeed likes to stick to membranes but doesn't like to go through membranes. And so nature has evolved some way, mechanism, to bring it in, from the extracellular into the intracellular fluid. This transporter is nicely blocked by a few compounds, one of which is this compound called AN404, of which we will talk a little bit more later on. Once inside cells, anandamide is a substrate for an enzyme which has been fortunately cloned. This is one of the few enzymes in this pathway of which we can say something at the molecular level, although I won't, because this is not our own work. Work done by Brain-Cravat at the Scripps cloned this enzyme. This is an enzyme that cleaves, it's an hydrolase, not very selective hydrolase, but very effective. It cleaves anandamide into arachidonic acid and ethanolamine, and also can be blocked by compounds such as this AM374 compound, fairly selectively. 2-AG inactivation is less well understood. Maybe there is also, in the case of 2-AG, a transporter involved. Maybe anandamide or AAH also participates in the cleavage of 2-AG. Or maybe there is this enzyme here, monoacylglycerol lipase, which has been described in several tissues, capable of cleaving this ester bond here, forming arachidonic acid and glycerol. And I won't talk very much about this. There's ongoing work in the lab indicating indeed that MEG lipase and not an N-amidahydrolase is predominant enzyme involved in 2-AG inactivation in situ, in intact cells. Okay, with this slide is put it there to remind me that I basically finished my first part of the presentation trying to wrap up what we know and what we don't know about the biochemistry of the endocannabinoid system. I'd like to move on the next portion, which is going to be more physiological and pharmacologically oriented. In the first series of slides, I'd like to present some data from our own lab suggesting a role for anandamide in the regulation of striatal neurotransmission and in the regulation of movement, as well as point to possible applications of cannabinoid-based agents in motor disorders. And I'd like to make 3 points, and the first of these 3 points is that anandamide is a striatal neuromodulator. So we base this conclusion on a series of experiments which were carried out in the lab by Andrea Giuffrida, and in collaboration with Larry Parsons at the Scripps Research Institute, in which we used macrodialysis in the dorsal striatum of rats. And what you see right here is basically a coronal section of the rat brain and a few tracks of our dialysis probes. And down here illustrates the method we have used. We've used gas chromatography mass spectrometry, or GC mass spec, to detect endogenously produced anandamide, and to quantify it by using an internal standard, a deuterated internal standard shown here. So anandamide is present in the effluents from the microdialysis probe and can be quantified. Okay. The first experiment we ran was aimed at determining whether anandamide levels extracellularly can be affected by standard things such as, for example, potassium chloride depolarization. And indeed, the answer to this is yes. And I apologize, the color is sort of faint. You may not be able to see it. But anyway, potassium chloride is right here. As you can see, this is anandamide level as percent control, percent baseline. And in the presence of potassium chloride, there is a significant, though not massive, but significant increase in anandamide release in 12 experiments. Now, the time course of this response, these are minutes, right? The time course is fairly slow, but as you can see in the inset, or you probably don't see in the inset, but let me tell you what is in the inset. What you see going up here is dopamine. And we basically analyzed dopamine in the same samples, showing that dopamine follows pretty much the same profile as anandamide, indicating that they are in a way, quote unquote, co-released. Don't take these words. Take these words with a grain of salt. I mean, released at the same time. By contrast, these compounds we saw before, palmitylethanolamide, PEA, and oleilethanolamide, OEA, are not being co-released, are not being released by potassium chloride. And also, quite interestingly, 2-AG, 2-alkylonylglycerol, is not released, and in fact is basically non-detectable. We see very small levels, almost undetectable levels in the dialysate. So it looks like anandamide is released somewhat specifically in the striatum. But before we make such a bold statement, let's look and see whether we can block this release with anything. And the first thing we looked at were obvious: tetrodotoxin, sodium channel blocker, and a calcium-free medium. And tetrodotoxin is pretty good in that, as you can see, this is control, this is potassium-stimulated, control, potassium-stimulated. In an artificial cerebrospinal fluid, we have this about 75 to 80% stimulation of anandamide release. TTX blocks it completely. Calcium-free creates some noise, but basically there is no effect in a calcium-free medium of anandamide, of potassium chloride on anandamide release. So this data suggests that anandamide is being released in a calcium-dependent fashion in the striatum, in the dorsal striatum of freely behaving awake rats, indicating that it's a striatum neuromodulator, possibly. The second point I would like to make is that there is something actually much better than potassium chloride to induce anandamide release, and that's activation of dopamine D2 receptors. Why did we look at dopamine D2 receptors? Because they are very abundant in the striatum. The striatum is a chock full of D1 and D2 receptors, and of course D1 and D2 receptors serve very major functions in striatal physiology, as well as in the control of motor activity, of movement initiation, and cognition as well. And the experiment I'm going to show you is very simple. We basically perfused quinpirole through the probe. This is a reverse dialysis approach. We did not inject it systemically. Quinpirole was perfused through the probe and anandamide measured. Now, if you look at, this is the levels of anandamide during quinpirole and after quinpirole stimulation. If you look at the y-axis, you can see that we have 900% stimulation, as opposed to the meager 175, 180% that we would get with potassium chloride. So this is a quite dramatic effect of quinpirole. Is it specific? Well, we believe it is specific because raclopride, first of all, because quinpirole is a fairly specific drug. Second, because raclopride, which is also a specific D2 antagonist, blocks the effects of quinpirole. Likewise, the compound SKF38393, which is a partial but fairly potent agonist at D1 receptors, is inactive in this regard. This is shown here. So the conclusion, I think the most economical conclusion of this data is that D2 receptor activation stimulates anandamide release in dorsal striatum. I'd like to qualify what I mean by D2 receptors. I mean D2-like or D2 family receptors. They come again in different subtypes or different types. They are pharmacologically undistinguishable or just become now pharmacologically distinguishable, the D2, D3, and D4. They can be presynaptic, postsynaptic. We don't know. We don't know much else about this phenomenon, except that we think that D2 receptors are tightly linked to anandamide formation in striatum. So the question is, what does this mean? What is the role of anandamide released by D2 receptor activation in the striatum? And our conclusion is summarized here. We believe that anandamide is an inhibitory feedback, countering dopamine-induced facilitation of psychomotor activity. And you take this, of course, with a grain of salt. It's sort of a simplification for the purpose of this presentation. What I'd like to convey to you is the idea that anandamide and dopamine, and D2 receptors in particular, are linked in a reciprocal fashion. And as dopamine produces a certain type of effect, anandamide activated by the dopamine system inhibits this effect as a negative feedback mechanism. How can we test this hypothesis? One way is fairly straightforward, is stimulating anandamide formation by, say, giving a dopamine agonist and blocking the action of endogenously produced anandamide using a cannabinoid antagonist. And this cannabinoid antagonist is shown right here, the compound SR141716A, which I will refer to as SR1 from now on. So one would expect, if indeed anandamide participates as a negative feedback to the effects of dopamine D2 receptor activation, one would expect the blockade of cannabinoid receptors to potentiate the effects of dopamine or a D2 agonist. And let's see what we find. These are experiments done in collaboration with Fernando Rodriguez de Fonseca at the University of Madrid. And in this particular experiment, rats were injected with different things, with vehicle, with quinpirole, the D2 agonist, or with the CB1 antagonist SR1, or with quinpirole plus SR1. And we were looking at different things, showing here only locomotion. This is horizontal locomotion. And as you can see here in the blank bars, the white bars, if you inject the vehicle, you have initially you have some kind of activity due to the injection, and then the animal stabilizes its behavior, its locomotor behavior. If you inject quinpirole, and it gets a little bit more complicated, but I'll walk you through it. If you inject quinpirole, quinpirole activates D2 receptors presynaptically and postsynaptically. The presynaptic D2 receptor is thought to be inhibitory on locomotor activity, whereas the postsynaptic is thought to be excitatory. So if you compare now quinpirole to vehicle, you have a nice inhibition of activity, locomotor activity. But as time passes and the concentration of quinpirole in the system increases, the D2 receptor gets activated, and you start to see a very nice stimulation of locomotor activity. This is thought to be a postsynaptic D2 receptor effect. If you inject the SR compound alone, it has absolutely no effect, and we have omitted it from this graph. But if you inject a combination of quinpirole plus SR1, this is what you get. You get a very nice potentiation, which is particularly strong at these points here, of the effects of quinpirole. And you may see the same, you will see the same thing regardless of how you do the experiment. If you look at locomotion instead of looking at immobility, the result is the same. And behavioral experiments being particularly tricky, I'm very pleased that this set of experiments have been confirmed by different laboratories. So we're pretty confident that this effect is indeed real. So one experiment was to block the cannabinoid receptor activated by endogenous anandamide. The other possible test of our theory that anandamide acts as a negative feedback is to test instead, is to do the opposite. Let's block anandamide inactivation. And anandamide inactivation, as I said before, is primarily regulated by this anandamide, this hypothetical anandamide transport, which we don't have as a molecule in a tube yet, but we know it's blocked by AN404. This compound was developed by us and Alex Macrianis at the University of Connecticut. we'll talk about it in a minute. But the idea is that if we give AM404, now we are blocking an anandamide inactivation. An anandamide produced by dopamine receptor activation should build up, and we should inhibit the effects of a dopamine D2 agonist. And let's see what results we got. Before I tell you about the results with dopamine agonists, I'd like to say a few more things about AM404. So that we all know what kind of molecule we are looking at and we are talking about. Here is the structure of AM404. It's very simple and it's very similar to anandamide. This is basically the paraphenyl, the para-hydroxyphenyl analog of anandamide. It produces a very nice inhibition of anandamide uptake in both astrocytes and neurons. And one can appreciate the selectivity of this molecule in a variety of different ways. And I've put here just an example. If you move the hydroxy group from the para position to the meta position, you quite dramatically change the potency of inhibition in astrocytes and the efficacy in neurons. Likewise, if you block this hydroxy group with a methyl, so you have the methoxy analog, that's inactive. And we have tested several, maybe close to 100 analogs of this compound with basically similar results. Changes in structure produce dramatic changes in pharmacology. We also know it's fairly selective in the sense that if we inject it into animals, we don't have a whole lot of things happening. In vitro, we don't. We tested it on 37 different potential targets, and we saw basically no effect. These targets include the CB1 receptor, the CB2 receptor, as well as the dopamine receptor. That's, of course, important for the next experiments I'm going to show you. One useful thing about AM404 is that allows us also to do a little bit of functional anatomy on the localization of the transporter, and this is by no means very high resolution data, and I'm sure anatomists in this crowd will be cringe their teeth at me, but I think it's as close as we can get to getting some idea where the transporter is. So these are experiments done by Massimo Beltramo in the lab, coronal sections of rat brain in the absence or presence of AM404, all of them in the presence of a cannabinoid antagonist, as well as radioactively labeled anandamide. So the antagonist impedes binding to the cannabinoid receptor, AM404 binding to the, impedes recapture by the transporter. So by default, we assume that this is predominantly, this staining is predominantly produced by, or this labeling rather, is predominantly produced by the transporter. And you can see that the striatum, the cortex, the factory cortex, the areas in the thalamus, the hippocampus, contain some detectable amounts of transporter activity. Again, this is not high quality, high resolution anatomy, but gives us an idea that indeed the transporter has some kind of discrete distribution in the rat brain, which is what we're hoping to see, in fact. So let's look at some pharmacology data. We have seen that AM404 is fairly selective. We injected it so with a fairly high degree of confidence by using ICV. And let me tell you that we injected ICV and we checked the concentration of the compounds afterwards, and the concentration that are reached never exceed 1.5 macromolar. And in this range of concentrations, we believe that the compound is, again, fairly selective. If we focus on the effect of AM404 injected by ICV in rats, in a test, in the apomorphine-induced yawning test, what is this test? Apomorphine is a mixed D1, D2 receptor agonist, but at the dose used in this particular experiment, which is 80 micrograms per kilogram, apomorphine predominantly activates D2 receptors. And the yawning response, though of course complex, is thought to involve the striatum. So we're looking at a fairly selective striatal response here, the yawning. And the number of yawns is computed here, is shown here, in the absence of AM404 or the SR1 compound. Now, if you give AM404 alone, you have a very nice inhibition of the yawning, the number of yawns. And this effect is completely blocked by SR compound. Now, I'd like to, the risk of sounding boring, which I might have already, I would like to point out that we don't believe in any effect of AM404 if it's not blocked by the SR compound. It's part of our pharmacological background to sort of double-check pharmacological experiments with an antagonist. So you can see here that there's a very nice inhibition of apomorphine-induced D1, D2, probably D2 activation. And we know that AM404 does not bind to D2 receptor, does not bind to cannabinoid receptor, so we conclude that the effect of AM404 is mediated through the release or the blockade of anandamide uptake. And here it's just shown that AM404 has the same effect, but in a dose-dependent fashion by given subcutaneously, and that anandamide has a similar response. Now, what about quinpirole? We've seen quinpirole before, and I think we should go back and look at quinpirole. I don't want to go over the whole set of data, but maybe we could focus on the immobility here on the back. Again, we are comparing multiple treatments, so please bear with me. I'll walk you through the experiment. We have vehicle, AM404, quinpirole, and AM404 plus quinpirole. So basically, the basic message is that AM404 alone increases immobility, that is, decreases movement, okay? And also it inhibits the activation, the motor activation induced by quinpirole, and we can see that looking at crossings and looking at immobility. This is fine and dandy, but it's drugs. We are giving basically D2 agonists, and we are looking at the effect of an anandamide uptake blocker. What happens in a model, in an animal model that more closely resemble human pathology? Now, the model, unfortunately, when we talk about neuropsychiatric disorders, I'm preaching to the choir here. We all know that there is no such a thing as a good animal model for basically any psychiatric disorder, at least not that I know of. But there is one strain of animals which is very interesting. It's called the SHR, spontaneously hypertensive rat, which was derived from a Kyoto strain of rats. And these animals are, of course, hypertensive when they grow older. When they are very young, they're juvenile, they have normal blood pressure, but they have a hyperactive behavior. In a way, they look like a type A personality. They get very active and then eventually develop blood pressure problems. We thought that this would be a nice model to test AM404 because we have seen that AM404 inhibits locomotor activity and inhibits dopaminergic actions, and there is increasing thought that the defect in the SHR juvenile rats could be produced by indeed dopamine hyperactivity, quote unquote. And please, when I say hyperactivity, don't read me as if I meant an increase in release or anything specific. It's just a hyperfunctionality, if you wish, of the dopaminergic system. So how do you do this experiment? These experiments were done in collaboration with Antonio Sadilla in the University of Naples. Basically what you do, you compare two strains of rats, the WKY rats and the SHR. The SHR are the pathological quote-unquote rats. And what you do is basically, we can focus on the lower one. What you do is basically two things. You put the rats in a cage and let them sniff around. They sniff around for, a normal rat will sniff around for, say, 15 minutes, then quietly slow down, eventually sniffing less. That's the end of the exploration phase. The animal habituated itself to the system. SHR rats behave differently because they always think to be in a novel environment. They keep moving around. And there are different ways you can look at this. And for example, if you look at the corner crossings, focusing first of all on the WKY rats and look at the orange bars first. These are vehicle injection. Corner crossings are higher in the first 15 minutes for the reason I said before, but then, they lower down, they come down. If you inject AM404 in these animals, nothing really happens. This is the green bar. With SHR, they move more. First of all, they are hyperactive by definition, and there is a small effect of AM404 on this, but it's not really dramatic. But what is really dramatic is in the last 15 minutes of the assay, you see the SHR rats are much more active in this second phase than the WKY strain they are derived from. And AM404 really completely normalizes their behavior. You can look at the rearing duration, which is the mirror image of this, and you will have basically the same type of phenomenon. So AM404 per se is not doing very much. You see, you don't see much of an effect of AM404 if you compare this or this. But in this pathological, quote-unquote, rats, it's normalizing their behavior. So this is very exciting, and we hope that in the future we'll be able to actually prove that this effect has something to do with dopamine and with anandamide. But it's definitely very, very interesting. So one way of summarizing this, and again, please take it with a grain of salt, is a working hypothesis is just that we think that dopamine, by activating D2-like, D2 family receptors, may not only have a positive effect on the psychomotor activity, that is lowering the threshold for psychomotor activation, which is one of the major function of dopamine, we believe, but also activate anandamide formation, which in turn, by activating cannabinoid CB1 type receptors, inhibits locomotor activity, assuring that the effects of dopamine is not either too protracted in time or too elevated in activity. A possible application of this in terms of disease is a series of conditions such as hyperactivity disorders and Tourette syndrome and possibly even schizophrenia, where there is general consensus that dopamine is dysfunctional one way or another. Do we suggest that we want to activate the cannabinoid receptor with, say, marijuana or hashish or cannabinoids? No, we don't. In fact, we are strongly against that possibility not for a political reason or an ethical reason, but for a scientific reason. We believe that activation of cannabinoid receptors with exogenous agonists will eventually lead to the desensitization of the receptor, and at that point there are many variables that we can no longer control. However, we believe that the possibility around this problem is by using indirect agonists, such as the AM404 compound, which blocks anandamide transport, or such as blockers of anandamide hydrolysis. In either case, we expect endogenous anandamide levels to increase and activate cannabinoid receptor in a much more physiological way in more restricted areas and in ranges of concentrations that more closely represent those present in a normally active brain. And possibly some of these conditions here might be in the future alleviated by molecules such as this. Now, in the last couple of minutes of my presentation, I'd like to shift gears and move from the brain to the periphery and describe another possible functional role of the cannabinoid system, endogenous cannabinoid system, in the periphery, and again, another possible pharmacological application. And we'll focus on the regulation of bronchial responsiveness. Although cannabinoid receptors are not very abundant in the bronchi, this is now work done in collaboration with Tamás Freund and István Katona in Budapest. István and Tamás were able to find cannabinoid receptors in peripheral nerve endings in the bronchi, and this is exemplified here on the top left of the panel. What you see here is a bronchial smooth muscle cell in the matrix. Right here is is a varicosity containing small vesicles, all these little guys here, and some larger peptide-containing vesicles, I suppose. I can't see them very well from here. But what you see here in dark is, as in this case, or in this case, or in these cases, are cannabinoid receptors as detected by immunocytochemistry using Ken Mackey's C-terminus directed antibody. And indeed, as you can see, the cannabinoid receptor, the staining appears to be in the inner side of the membrane, of the plasma membrane, corresponding to the localization of the antigen to which the antibody was directed. So these data suggest two things. The first is that there are cannabinoid receptors in the airways, and the second is that they are restricted in prejunctional sites, which is very much quite interesting, in my opinion, and also reflective of what happens in the CNS, where the cannabinoid receptors are very abundant, I wouldn't say exclusively because that's probably wrong, but very abundant in presynaptic nerve terminals. So, I'm sorry. So we have cannabinoid receptors in the lungs. Do we have anandamide? The answer, of course, is yes. Otherwise, I wouldn't be talking to you right now. but we can measure anandamide biosynthesis using a technique similar to GC mass spec. It's called high-performance liquid chromatography mass spectrometry, HPLC mass spec. And again, it's based on the same principle. This is basically endogenous anandamide, and this is the internal standard, the deuterated internal standard. It's present, but that's not such a big surprise. But what I think is very exciting is that biosynthesis of anandamide can be stimulated by calcium. Calcium, of course, is a primary mediator of a number of neurotransmitters and neuropeptides in the lung. And this is appreciated here, where calcium was found to stimulate to triple the levels of anandamide with respect to an EGTA incubation where calcium is chelated. Together with anandamide, the anandamide precursors, you may or may not remember from the first part of my presentation, I talked about this NAPES, precursor of anandamide, N-acyl phosphatidylethanolamin's, and they actually come in different families. It's a kind of a complex family of molecules, and two of them in the lung are shown here. For those of you who are inclined towards lipid chemistry, this is an alkenyl ether, and this is an alkyl ether. We find that the only precursor for anandamide are alkyl or alkenyl ethers and contain a very nice arachidonic acid group here, which we identified again by HPLC MS. And we could quantify that, although not by using isotope dilution, this is in the case of anandamide, but we can quantify it, and this is the compound B1, the tracing B1 refers to compound A1, the tracing B2 to compound A2, and you can see that calcium really dramatically increases the synthesis of these compounds together with the synthesis of anandamide. That makes sense because anandamide and its precursor are produced in concert. As anandamide is cleaved from the precursor, N-acetyltransferase activity kicks in and synthesizes more anandamide, and synthesizes more precursor to form more anandamide. So we have the whole machinery in the lung, and what is the function of this machinery? We don't know, of course, but we have some ideas. And one of the ideas that we have is that may regulate bronchial responsiveness. And I won't show you the whole complexity of the data. I'll try to simplify it, showing in particular the fact that anandamide inhibits bronchospasm induced by a compound called capsaicin. Capsaicin is to the pharmacologist, the vanilloid receptor agonist. To the non-pharmacologist, is the active principle in chili pepper. It's, of course, very painful compound. And when guinea pigs inhale capsaicin, they cough. And in fact, you can see they cough in a dose-dependent fashion. This is capsaicin. This is injected, actually, but inhaled would be the same. Capsaicin produces nice cough response. Nice bronchospasm. We'll talk about cough later on, right here. Increasing bronchospasm, and this is blocked by capsaicin, which is a vanilloid VR1 receptor antagonist, a fairly selective antagonist, although deserves improvement. But this is, I think, enough to show that this is a vanilloid-like receptor effect. And the effect of capsaicin is very nicely inhibited in a dose-dependent fashion by anandamide. These are doses in milligram per kilogram delivered IP. And you can see at the highest dose of 5 milligram per kilogram, anandamide completely blocks the bronchospasm induced by capsaicin. Now, let me say one word about the mechanism of capsaicin. Capsaicin is thought to act by activating, as I said, vanilloid receptors, which are present on terminals of sensory terminals in the lung. What capsaicin may do is it may stimulate release of neuropeptides, which in turn produce the bronchospasm. So it does stand reason, the fact that anandamide is inhibiting the activity of capsaicin, because if anandamide receptors, the cannabinoid receptors, are present presynaptically and they are inhibitory, this is what you would expect. And I think this is the likely mechanism of action of this particular response we are observing here. Just to make sure that we are talking about a real cannabinoid receptor, we used the compound SR1, SR14-17-16A, the CB1 antagonist. And as you can see, not only does SR1 block the effect of anandamide, it kind of overshoots, which may also due to its inverse agonist properties. By contrast, the compound SR14-4528, or SR2, which is a CB2 antagonist, at a dose of 0.5 milligram per kilogram, does not really inhibit the effects of a maximal dose of an anandamide. There is a little bit of inhibition, but frankly, I think this probably is due to the overlapping of SR2 with the CB1 receptor. It's not a completely selective compound. And not only does anandamide inhibit bronchospasm, it also inhibits cough, which is very exciting. It does so by IP injection, as shown here. This is anandamide by IP. This is the number of coughs produced by capsaicin, aerosol capsaicin in a guinea pig. But if you give anandamide by aerosol, the effect is even stronger, which again suggests that the effect of anandamide are peripheral. They are right in the upper airways where the cough reflex is started. Again, this is blocked by the CB1 antagonist, but not by the CB2 antagonist. So what we think the role of anandamide may be in the lung is, again, a role of a negative feedback. When, say, an irritant stimulates cough or bronchospasm, release of anandamide may alleviate that, sort of a push-pull or yin-yang type of phenomenon. And pharmacologically, I think this is interesting because there aren't very many compounds that inhibit cough, particularly chronic and productive cough. And again, this offers a possibility in that direction, which is worthwhile exploring. And well, this is one piece of data I forgot about, I'm sorry. It just points out to the fact that if you give capsaicin alone at a dose like 3, and look at bronchospasm or the number of coughs, a low dose, which produces non-complete bronchospasm, incomplete bronchospasm, then if you combine that with SR1, you get a very nice potentiation, which implies, although not completely, but it does suggest that indeed activation by capsaicin releases anandamide, and anandamide now engages the cannabinoid receptor. We block it with SR1, and we get more bronchospasm because anandamide is impeded to do its normal job, which is to inhibit the bronchospasm. The same thing here with cough. So finally, I'd like to thank all the members of the lab who have participated in this study. Andrea Giuffrida he did the HPLC-MS work in striatum, and a lot of the other people in the lab now are doing new projects, and hopefully if I ever get to talk to you again, maybe I'll thank them for some of their own contributions, but they haven't really contributed to any of the specific projects I talked to you about today. Thank you, and I'll be glad to entertain any questions you might have at this point. Yes. The question is, do you have any idea as to how D2 receptors are linked to anandamide synthesis? And the answer is no. Actually, we don't. An experiment to do would be to take cells in culture and study the mechanism using cells in culture. And we just have not done that. But it would be important to know, yes. Yes.
I was wondering where you think anandamide is being released from in the striatum.
That's a very important question, I think. The question is again where from is released in striatum. We can answer a little bit more on this particular question because we did culture striatal neurons, medium spiny neurons predominantly, and these neurons are capable of producing anandamide. They do produce a substantial amount of anandamide stimulated with, say, ionophores or potassium chloride or kainate. They also take up anandamide quite avidly. So the medium spine neuron has the whole machinery, at least in culture, the whole machinery to produce and inactivate anandamide. In vivo, could be the medium spine neuron, could also be the presynaptic ending, or could be some associated, for example, presynaptic ending coming from the cortex, as well as those coming from the substantia nigra. We really don't know. My guess, unlike the first question, my guess is that the medium spine neuron is the most likely candidate, but we have no direct data to prove that.
So I guess the question is whether or not the quintrol release or induction of anandamide release is activity dependent. Did you try that?
Yes, it is not. Actually, tetrodotoxin does not block the effect of quinpriol, and vice versa. Raclopride does not block the effect of potassium chloride. That much we did. Yes?
I'm a little confused about how you think this functions as an anandamide. Would it be like a classical co-transmitter like ATP, or do you see this as something that's kind of an elaboration of the icosanoid pathway? It certainly seems, and perhaps related to CPLA2 activation.
No, I don't think it's an elaboration of the. I don't think it's either, as a matter of fact. I think it's its own independent system that has evolved probably independently of the icosanoid system. The icosanoid system is based on a phospholipase A2 forming a free arachidonate, which is then metabolized. We have no phospholipase A2 here. The free arachidonate comes from the SN2 position, and in this case, the anandamide comes from an N-linked position. Instead of a PLA2 is a PLD. There is a net N-acetyltransferase, which is not involved in the arachidonic acid pathway at all. It's a lipid pathway, but it's distinct from the arachidonic acid pathway. It does have some formal homologies with that, insofar as all lipids, must come from somewhere in the membrane, and so that there is some sort of formal homology there, or analogy rather.
This is just a synthetic pathway that you see for its formation.
Right. This is synthetic pathway based on a number of experiments. We believe that this is more or less the way these compounds are produced. Yeah.
And with respect to the PLD, there are three that I know of whose structures are known. One which is related to the PI anchor, and two which are probably PC specific. Is this a fourth one? Have you tested those activities to know if they?
Okay.
There's also an activity that's PE specific, but as far as I know, nobody's purified that.
The PE-specific activity is not purified to my knowledge. The PC activities have been cloned, of course, yes. We call it a PLD based on the fact that in enzymology, any enzyme that cleaves the distal phosphodiester bond is a PLD. It's a phosphodiesterase, D-type phosphodiesterase. So from a nomenclature standpoint, we are okay. But if you ask me whether we know the structure, we don't know the structure. No, this is an unknown enzyme. And that also...
It's very like the base exchange pathway.
Could you say that again?
The calcium dependence is like the base exchange pathway, which is something that...
Yes. Well, thanks for asking that, because in fact the calcium dependence here is created not so much by the PLD reaction, by the N-acetyltransferase reaction. See, the two reactions are really together. They're really together. It's almost impossible I would say I never succeeded in detecting release of an anandamide without stimulating the precursor for an anandamide. The two things are really hand in hand, and Sugiura in Japan believes that the key step in an anandamide formation is actually NET, not PLD. And I actually agree on that based on no data, but Sugiura's data suggests that.
You still have to have the arachidonic acid.
You need to have arachidonic acid on the N position, and that arachidonic acid comes quite surprisingly from the SN1 position of phosphatidylcholine. There is a small, about 0.5% amount of phospholipid in brain that contains arachidonic acid in the SN1 position. We detected that. We published in Neuroscience 97. And we believe that that's the precursor for the precursor of an anandamide. Yes?
The pharmacological profile of the bronchospasm looked like it was mediated through the CB receptors, but I'm wondering if you can comment on the potential for anandamide to act as a capsaicin analog for agonists.
Yes. Of course, this has attracted a lot of attention, and I believe that there is no reason to think that a vanilloid receptor is involved in these actions. For one, the effects of anandamide are opposed to those of capsaicin, and I may be a dumb pharmacologist, but it doesn't strike me as logical that you're activating a receptor which normally produces bronchospasm, and depending on how you activate it, you get bronchospasm or bronchodilation. I mean, I'm aware that there are ways receptors can couple to G proteins that are different, but to the point of exerting radically opposed effects, it's a little bit hard to swallow. So in terms of macropharmacology, it doesn't make sense. But also in terms of the type of blockers that block the response to capsaicin and those that block the response to anandamide, we have, again, a pretty standard situation. Capsaicin is blocked by capsaicin, and anandamide is blocked by SR1. And that's about it. SR1 potentiates the effects of capsaicin. So then again, we see no overlapping. Again, we see something that is more parsimoniously explained with a negative feedback system than with an activation or co-activation of the same receptor. Yes?
In the structure of the precursor, you have the portion that is become anandamide associated with the membrane. But yet it seems to get released so that you can pick it up by microanalysis. How do you envision the transporter as being involved in taking it out of the membrane and releasing it into the aqueous media?
We tested that hypothesis. Let me just rephrase for everybody. The hypothesis, the question is, what is the mechanism for anandamide release? And of course, it's a very important question. Let me just put it in the context of these lipid signals. It's a question that applies to basically all lipid messengers, leukotrienes, prostaglandins, they all, and now anandamide, they all have the same problem. They are produced often in the cell, and indeed, for example, leukotrienes are produced prostanoids are fairly hydrophilic, are produced on the inside the cell at the level of the cytoplasmic reticulum. How these things go out, the general thought in the field is that there are transporters. In the case of leukotrienes, for example, a leukotriene exporter has been identified. It's kind of an ATP driven transporter of the flipase type that participates in the flipping of phospholipids. In the case of anandamide, we don't know. We have tested the possibility that the anandamide transporter, which internalizes anandamide, participates also in the extruding of anandamide, and our experiments tend to rule out that possibility. And one reason is that AM44 doesn't block anandamide formation, anandamide release, and also when we try to load neurons with anandamide, basically anandamide gets immediately hydrolyzed by the anandamide aminohydrolase. So as soon as anandamide is inside the cell, is the target of the aminohydrolase that cleaves it. So our current model is that perhaps anandamide is produced in the membrane, and either there is a chaperone protein that helps, like a lipocalin, for example, that helps moving around, or there is indeed a transporter that kicks it out into the medium. We don't know. Yes, George.
In the early part of your talk, you had this intriguing observation in this triangle stimulation model that you got nice anandamide release but no 2AG.
Mm-hmm.
We recently found and published that in an intravenous injection in mice, 2AG is extremely rapidly inactivated, disappears within seconds, whereas in the same paradigm, anandamide survives for minutes. And that suggested to us that it may be a monoacid glycerol lipase that is responsible for the unique degradation of 2AG. Have you considered the possibility that maybe it's degraded before you can detect it in the system?
Yes, yes, it is. That's possible. That is possible. In general, we don't find 2AG, I must say, associated with fluids. For example, in cerebrospinal fluid, we see anandamide. We don't see 2AG. In dialysates, we see anandamide. We don't see 2AG. I'm not sure that it's not caused by a degradation difference. Another possibility is that 2AG is not very effectively released, whereas anandamide is, which could be interesting. You know, we could still have 2AG formation without 2AG release, and that would be a true autocrine at the cellular level, a true autocrine signal at the cellular level. Difficult to test. Yes.
What is the concentration range of the anandamide in the extracellular of the striatum, and what kind of method you use to measure it? And second question is quite like the dopamine D2 receptor and the serotonin 2A receptor share the similar transduction pathway. For example, serotonin 2 receptor activates the G protein, then coupled to the PRC enzyme. Do you have some information to know serotonin 2A receptor also activate the release of anandamide?
Okay, the second question is, do we know anything about serotonin receptors in the activation of anandamide release? And the answer is no, so that's easy. The first one is a little bit more complicated. You're asking whether we know the extracellular, the interstitial, I suppose, concentration of anandamide by microdialysis. As you know, to do these are very complex measures, and we have not done those measures. Ideally, one would like to be able to measure these things at where they act, at the site of action, and having setting up the whole methodology to measure them in interstitial fluid, knowing that that is not even where we really need to know their concentration, sort of demotivated us, and we decided that maybe we'll do it some other time when we have a more high throughput assay for doing anandamide. See, this is one thing that I'd like to say to honor my colleague Andrea Giuffrida, who is now an assistant professor in the lab, who was a postdoc when he did these experiments. Each assay is approximately 60 minutes in length. He analyzed 75 rats, each of which had 45 samples. So the maths is easy. It's a lot of work, and sometimes you have to. When he was promoted, he negotiated that he wouldn't do it again. Yes.
Can you measure that lipid precursor?
Yes. Yes. We show that in the bronchial data. Yes, we measure the precursor.
And is it there in concentrations lower than like 53? How much of the total lipid is?
Oh, how much of the total lipid? That's a good calculation. I've never done that. We measure picomoles per milligram of protein. We don't usually express per lipid, per quantity of lipid, but I think you're right. That would give us an idea of the sort of the concentration in the membrane, and that's a good exercise. Yeah, we should do that with phosphorus measurements, for example.
Would you expect it to respond to something like CTPLA2 inhibitors? Concentration. I mean, from your model, it sounds like you wouldn't expect.
Okay, we studied the pharmacology of the NAT enzyme, and of course, what is NAT? NAT is an N-acyltransferase. It takes a fatty acid and it transfers it. So initially, it has a lipase reaction, right? So it has a lot of molecules that block phospholipases tend also, those most non-specific inhibitors tend also to block NAT because they block the first step in which the SN1 position is where arachidonic acid in PC is sitting, is cleaved, and then arachidonic acid is moved. The first position is, the first reaction is blocked. So, for example, bromophenacylbromide will block NAT. A number of boron compounds will block NAT. But anything specific, we don't have any specific inhibitor. We haven't even tried yet, although we do test a whole lot of different compounds in the hopes of finding selective blockers of various pathways. We haven't focused yet on NAT because we're focusing on its molecular characterization right now, not so much on the pharmacology.
Well, to interrupt this very interesting debate, but some of you here are regulars, so maybe you can tell me I'm relatively new, that I saw some tables with food preparation. If that's for us, I would suggest that we continue the discussion in an informal manner outside. Thank you very much.
Thank you.